In this video, we're going to continue our discussion of gluconeogenesis. So where we left off is that we made oxaloacetate from pyruvate, and that is made in the mitochondria. Now, the rest of the steps of gluconeogenesis, converting PEP to glucose, that is done in the cytosol. Now, making PEP itself, where that takes place, can depend on the organisms. Humans, you can make it either in the cytoplasm or mitochondria. But at the end of the day, you have to move either oxaloacetate or PEP from the mitochondria to the cytoplasm to do gluconeogenesis. Now, if it's PEP, that's no problem. Mitochondria has a special transporter for PEP, and you can just move that to the cytoplasm. Oxaloacetate is more difficult though. Oxaloacetate does not have a dedicated transporter, so you have to do a more convoluted transport system to get oxaloacetate into the cytoplasm. And that is, you have to convert either oxaloacetate either to aspartate or to malate before you can transport it out. So we have two different routes. We're going to look at route 2 first, that is converting it to malate. You convert oxaloacetate to malate. And this is done using the enzyme malate dehydrogenase. And what you do is that oxalate goes to malate, and you use NADH to do this. Malate can then be transported into the cytosol. Malate can go back to oxaloacetate, and you remake NADH. So all you're really doing in this whole mechanism is that you're moving oxaloacetate out to the cytoplasm, and you're also moving NADH from the mitochondria to the cytoplasm as well. Now, the aspartate path is a little different in that it does not involve NADH at all. And so you don't really use this path under most conditions. And the reason is, when you do gluconeogenesis, you need to use NADH. So, if you do the malate pathway, you're making NADH in the cytosol that can be directly used for gluconeogenesis. If you go the aspartate pathway, you do not make NADH, so you're missing something from gluconeogenesis. However, if you're using lactate for gluconeogenesis, lactate will make NADH in the cytoplasm itself, so you can use the aspartate pathway. And so the aspartate pathway is oxaloacetate goes to aspartate via aspartate aminotransferase. You get an amino group from some amino acid, and you're left with an alpha keto acid. Aspartate can now go to the cytosol. Aspartate will do the same reaction again. It will give its NH3 up to an alpha keto acid to form some amino acid, and then you'll be left with oxaloacetate. Note that all of these reactions are reversible, and you can use pathway 2 to transport NADH into or from the mitochondria. So path 2 you can just use if you need NADH to go to the cytosol, just use this pathway. Run it in reverse if you want NADH to go from the cytosol to the mitochondria. So that covers really the first two steps of gluconeogenesis. The next alternative enzyme we run into is fructose 1,6-bisphosphatase, FBPase. And what this is going to do is going to hydrolyze a phosphate from fructose 1,6-bisphosphate to make fructose 6-phosphate. Fructose 6-phosphate is then isomerized into glucose 6-phosphate. Then glucose 6-phosphate will lose a phosphate via glucose 6-phosphatase. Note, we already ran into glucose 6-phosphatase. This is the enzyme that converts glycogen glucose 6-phosphate to glucose. And this is only found in your liver and kidney. Note, to go backwards, you need to burn 6 ATPs to change 2 pyruvates into glucose. Going forward, breaking down glucose to 2 pyruvates, you only get 2 ATPs. So if you just do this cycle over and over and over again, you lose 4 ATPs. And note, it is possible to take some of these intermediates and shuttle them off to different pathways. For example, the pentose phosphate pathway that we talked about. Now, since we're losing 4 ATPs if this cycle just runs in a futile way, We don't want that to happen. So gluconeogenesis and glycolysis are reciprocally regulated, which means you turn one on while turning the other one off. This happens at 3 key points, 3 substrate cycles that we already just talked about. And flux from PFK and FPBase, which is our second substrate cycle, which again, remember, is the rate determining step of glycolysis. Well, that's determined by a molecule, fructose 2,6-bisphosphate. Note that we usually use fructose 1,6 in glycolysis. Here we're using fructose 2,6 for inhibiting and turning on an enzyme. Now, fructose 2,6-bisphosphate will activate PFK, so it will activate glycolysis, and will inhibit FBPase, inhibit gluconeogenesis. And the balance of how much of this you make depends on the synthesis and degradation by phosphofructokinase 2, PFK2, and fructose bisphosphatase 2, FBPase 2. These are 2 quote-unquote enzymes. And I say quote-unquote because it's actually, these 2 things are one enzyme. PFK2 and FBPase2 are the same enzyme. They're just different domains of the same enzyme. And if you notice down here, if we have fructose 6-phosphate, if PFK2 is activated, this will go and make fructose 2,6-P, which will turn on glycolysis. While if FBPase2 is activated, that will take away a phosphate from F26P, will make F6P. Now glycolysis is inhibited and gluconeogenesis is activated. So let's just look at the logic for this. So let's say you're at low blood glucose levels. You want to increase glucagon. You want to tell your body, hey, glucagon is gone. And so we looked at that pathway already. Glucagon can go bind to the receptors in your liver. Then your liver will release cyclic AMP as a signal to say we're low in energy. Cyclic AMP then will increase enzyme phosphorylation. Because remember, cyclic AMP turns on pKa. pKa will then turn on or phosphorylate other enzymes to turn on glycogen breaking down. This will also activate FBPase2 and inactivate PFK2. So this will turn on gluconeogenesis because we're out of glucose, start making glucose. Don't go through glycolysis. We don't have a lot of glucose, so don't break down glucose. This will decrease F26P, so this will inhibit PFK and activate FPBase, increase gluconeogenesis. So you can see everything we've talked about in this chapter in metabolism in general is all tied together. So it seems, and it is actually very overwhelming when you first learn of it, but just don't lose sight of that it's actually all connected and it's very beautiful logic. Low blood glucose, start making glucose. The opposite happens at high blood glucose. Stop making glucose, start breaking down glucose. Now, there are other infectors that influence gluconeogenesis. Pyruvate kinase can be inhibited by alanine. Alanine is actually a major precursor for gluconeogenesis. Alanine can be converted to pyruvate if you just remove its amino group, and this is transamination. So as seen here, if you just remove NH3, bam, you got pyruvate, you can start making glucose. Liver pyruvate kinase is inactivated by phosphorylation. This can also activate glycogen phosphorylase, as we talked about in a previous video. However, if you want long-term changes to enzyme synthesis that affects the overall flux through either glycolysis or gluconeogenesis, you want to change the amount of enzymes you have available. So your pancreatic and adrenal hormones can influence the rate of transcription and stabilization of mRNAs. And if you remember your central dogma of biology, mRNA will go to the ribosome to make protein. So if you make your mRNAs more stable and you make more of them, you're going to have more enzymes. Note insulin will inhibit transcription for the gene PPCK, not PEC, while CAMP promotes PPCK, FPBAs, and glucose 6 phosphatase being made. So you don't have any energy, make these genes. While insulin says we have a lot of glucose, don't do gluconeogenesis. We don't need it. And that's it for our chapter on glucose and glucose metabolism. So I hope you had fun through that journey. And in the next chapter, we will look at the citric acid cycle. Until then, I guess I'll see you then.